It is our great pleasure and honor to introduce Dr. Fried to the keynote lecture tonight. I have 3 pages, which I will not go through them. Don't be worried, but I did my homework. So Dr. Fried got his PhD in 1990 in the laboratory of Dr. Rex Reeser and Howard Temin at the University of Wisconsin-Madison. And then he, in 1991, he moved to do a postdoctoral training with Dr. Temin there, where he worked on MLV and HIV envelope glycoprotein in fusion and entry. He then moved in 1992 to the Laboratory of Molecular Microbiology at the NIAID, working with Dr. Malcolm Martin on HIV assembly, entry and post-entry events. In 1997, he became an investigator there, and in 2003, as you know, he joined the HIV drug resistance program. And he became the director of this program in 2015. He has been coming to this meeting at Cold Spring Harbor for the last 37 years. And he actually organized this meeting 20 years ago in 2004. Yeah, there are so many hours here. I should select a few. I hope that he will agree with the one that I selected. And so he got the NCI Mentor of Merit Award in 2010, the NCI Women Scientist Advisors Mentoring and Leadership Award in 2021 for the excellence in mentoring, guiding careers of scientists at all stages of their careers. I think it's extremely important. In 2018, he obtained the KT Jing Retrovirology Prize, and he was selected to fellowship in the American Academy of Microbiology in 2019. I will not go through all the papers. Suffice it to say that he made seminal work in all the best journals and at all the stages in several topics on assembly, release of HIV and other retroviruses, which is kind of obligatory in my lab to read to become a member of my lab. Envelope incorporation and function and virion maturation. So without further ado, please join me to welcome Dr. Fried.
Well, thank you very much, Andres. Is my mic on? All right. Check it out the wave. Okay. Thank you, Andres and Florence, for the very kind introduction and the invitation to give this lecture. I'm especially humbled to be giving this after Judy Liebern's really fantastic talk the other evening. Let me get started. So this is, if not quite an outline, but sort of a series of threads or themes that I'm going to touch on tonight. My personal and professional journey, what this Cold Spring Harbor Retroviruses meeting has meant to me in my career. I've basically been coming here my entire adult life. I'll say a few words about service to the community, and I'll say a little bit about what we're studying now. So there will be some actual science in this talk. So I grew up in the Appalachian Mountains of central Pennsylvania, which some of you might know is a very beautiful part of the country. And as a result, I became very active in outdoor activities. And here's nerdy little Eric, winter camping and ice fishing and backpacking. My dad was a physics professor at Penn State University. So because of the fantastic tuition break that I received, I went to Penn State as an undergraduate. And I was initially a biology major, expecting to do some kind of field biology that would take me to the outdoors. But really the epiphany for me was when I took organic chemistry and biochemistry. I know that sounds weird because I think most of you probably hated organic chemistry. I really, sort of became determined to learn about the molecular underpinnings of biological systems. And so I changed my major from straight biology to what was then a brand new major, molecular and cell biology. There were only 18 students in this major at Penn State, in contrast to hundreds of biology majors. And so through my coursework, I became really fascinated by viruses, initially these very elegant mechanisms of gene regulation in bacteriophage. And I also became really intrigued by work coming out of the Varmus and Bishop lab on transforming retroviruses and the oncogenes that they carry. So as an undergraduate, I did research in David Shalloway's lab, working on a cooperation between oncogenes. And this was actually not quite my first paper, but it was the work that I'd done as an undergraduate. I then went to University of Wisconsin-Madison, which at the time and still I think is sort of a mecca of retrovirology. This is the McCardle lab where I worked before a multi-story parking structure was put up in front of the building. And I loved to cross-country ski, so Madison was a great place for me. And this is the Isthmus of Madison. This is the Monona Terrace where the ASV meeting is held when it's there. And in Madison, I worked in the lab of Rex Risser, a young professor who is well known for studying Abelson murine leukemia virus and transformation by that virus. And a couple notable individuals who were associated with Rex's lab. Pat Green, now at Ohio State, was a senior graduate student in the lab. And Jackie Dudley had already left the lab when I joined, but she was credited by Rex for having brought molecular biology to the lab. So we really all benefited from her former presence in the lab. And so I remember my first day in Rex's lab. He sat me down in his office and he said, "So, what do you want to work on?" And I thought, well, you're my advisor. Aren't you supposed to tell me that? But I didn't say that. And fortunately, I spent the Christmas break reading the thesis of one of the former members of the lab and was really intrigued by a description of a pair of murine leukemia viruses that differed by only a few amino acid residues, but one of them was highly infectious and the other was very poorly infectious. And to make a long story short, the difference between these two I mapped to the cleavage site between GP70 and P15E, the envelope glycoprotein precursor. And so this work demonstrated that cleavage of PR85 is essential for virus infectivity. This seems really obvious to us now, but it was actually not known at the time. As Andres mentioned, my first year here was 1987. I was 23 years old. And this is the cover of that abstract book. It's somewhat primitive. And this is the first abstract, which was a poster. And at that meeting, and at that meeting and the many meetings that followed, I realized that this was a place where I was really going to get to know my colleagues who would be important players in my career. Here I'm showing those who became my close friends and colleagues in the DRP, which, as you know, I joined much later on. John, Steve, Alan, and unfortunately David Dursey is no longer with us. So this was in the early years of the HIV pandemic, and so as a young retrovirologist, I switched my focus from murine leukemia virus to HIV. This was not without a few speed bumps in the sense that no one in Rex's lab, or for that matter at UW-Madison, studied HIV at that time. So I really had to obtain or make all the reagents that I needed for my work. But nevertheless, I switched to HIV but continued an interest in envelope glycoproteins. My first project on HIV envelope was to characterize the cleavage sequence between GP120 and GP41, that is the GP160 cleavage sequence. And so I came back to Cold Spring Harbor, and I promise I'm not going to go through every one of these. I came back in 1988. This was the cover of the abstract book, and in stark contrast to this year's abstract book with these beautiful, sophisticated cryo-EM structures, here we have, kind of our view of retrovirus replication at the time with this little cartoony virus. This was both the front and the back cover. So whoever designed this, I think, was really stretching for something to put on the cover. The organizers that year were Steve Goff and Naomi Rosenberg. And this was my first talk, the first time I stood at this podium, and I was terrified. Well, okay. But what's sort of interesting is in those early days, the HIV session was its own session. So it's kind of fun to look at the people who were presenting or were authors of abstracts in that session. We have Eric Cohen and Bill Hazeltine Klaus Strabel, Malcolm Martin. Klaus is probably here, I think, somewhere. Vinay Prasad, Steve Goff, Eric Hunter, Beatrice Hine, George Shaw, and so on. So I continued to work on HIV envelope, characterizing the fusion peptide of GP41 and showing that the V3 loop of GP120 is a domain important for fusion and infection. And we actually sent this paper to Science, but the people at the front desk said, I don't think there's really any interest in the V3 loop. So the paper was summarily rejected. I think there have probably been many thousands of papers on the V3 loop since, but anyway. Much of this work relied on the fact that expressing HIV envelope in CD4 positive cells led to the formation of these multinucleated syncytia. So these could be scored microscopically, and here I am looking through the microscope scoring these syncytia. This is a little dish that has syncytia on them, and this is back when I had a lot more hair on the top of my head. So really quite tragically, my PhD advisor, Rex Risser, died in 1990, just several months before I was to defend my thesis at age 42. So Howard Temin took me under his wing, and it was really a great time in that lab. A number of notable individuals were in the lab at the time. So this is me and Wei Su and Winnie at the Wine and Cheese party here at Cold Spring Harbor. Lou Mansky, and here we are, myself and Lou, talking with Alan Engelman over a beer and a glass of wine. This is Wes Sundquist, I believe, and I think this might be Paul Binoche. And back here is a very agitated Jonathan Stoy. And Kathy Borislari was also in that lab at the time. I don't think she's here this year, but interesting little story about Kathy. So during this period, this meeting went from Tuesday to Sunday. And Kathy's husband called the meetings office and was very irate that, why do all these scientists have to, kill their Memorial Day weekend year after year? And so meetings office person came in and said, let's take a show of hands. How many of you want to move the meeting from Tuesday to Sunday to Monday to Saturday? And everybody said yes. And this is why we have maybe I'm getting a few details wrong, but, artistic license. But anyway, you can thank Kathy and her husband for having Sunday off. And that part I know is true. I'm up to 1990, guys. We're making progress. So 1990 was a very important year for this meeting because it was the 20th anniversary of the discovery of reverse transcriptase. So the two keynote speakers that year were Howard Temin and David Baltimore. And here's Howard having his piece of RT birthday cake, and Howard Temin, David Baltimore, and John Coffin enjoying a glass of champagne afterwards. So my last paper in Madison showed that a mutation in the fusion peptide of GP41 exerted a dominant negative effect on membrane fusion. This work suggested that HIV-1 envelope must be a multimer. And of course, we now know that the HIV envelope and all retroviral envelope glycoproteins are trimeric. So in 1992, I went to the NIH, NIID, Bethesda, to work with Malcolm Martin. I continued to come to this meeting, so here I am having a beer with Federico Romani. One gets the impression from looking at these photos that all we do here is drink. But there were actually, talks like this even back then. So I continued to study HIV, but I shifted my focus from virus entry and membrane fusion to primarily virus assembly and release, looking at the role of matrix and GP41 in envelope incorporation, some work on characterizing the role of P6 in virus budding, and also waded into the, even then 25 years ago, controversial topic of HIV nuclear import, showing that matrix is not the determinant of nuclear import as had been thought at that time. My son Ben was born in 1996. He's now a PhD student at Carnegie Mellon in computer science. My daughter was born the next year, 1997. Here we are is my lovely wife, Janelle. My little baby, Tasha, just graduated from medical school on Sunday. And here I am drinking again. This is really going to damage my reputation. Okay, so in 1997, I got a tenure track position in NIH NIID. We continued to work on envelope glycoprotein incorporation into virus particles. This was work by Tsutomu Murakami. Akira Ono, who's sitting right there, did some very important work showing that HIV assembly takes place in lipid rafts and that PIP2 is an important determinant in GAG trafficking to the plasma membrane. And we also contributed to the discovery that the escort machinery is recruited by P6 to promote virus spreading. Other very notable players in this area were Paul Binash, Carol Carter, and Wes Sundquist. I received tenure in NIID in 2002, and then in 2003, moved my lab to what was then the HIV Dynamics and Replication Program, since renamed, the HIV Drug Resistance, I get it messed up. It was then the HIV Drug Resistance Program, now the HIV Dynamics and Replication Program, and you can see in the front row a number of very familiar faces. And we are making progress. And in 2004, 20 years ago, I coordinized this meeting with Susan Ross. And, you know, a lot about this meeting has changed. People have come and gone. But one thing has remained constant. Yes. Okay. I'm going to say just a few brief words about service to the community, which I think is very important. This is a bit of maybe a touchy issue for me because I always feel like I'm involved in way too many things, particularly editorial things. The most time-consuming of these has been serving as editor-in-chief of Viruses, which I've been doing since the founding of the journal in 2009. And being editor-in-chief of a journal is kind of a thankless job. Sort of like being a high school principal. You only hear about things when there's a problem. An example of this was this expression of concern that Oliver Schinglin and I published on a paper that claimed that SARS-CoV-2 spike impairs DNA damage repair. This paper was later retracted, and this whole episode drew a lot of attention from the anti-vaxxers because they used this paper as evidence that the SARS-CoV-2 vaccine was dangerous, and this created a bit of a firestorm on social media, including this particular tweet that Alex Kleinpeter in the lab showed me, much to his amusement. I made a picture of them. Shilgin is the one that looks like Jack Black meets Mr. Potato Head. I love how it ends. Sorry, but it's true. So despite these little setbacks and embarrassments, I do believe that service to the community is an essential part of what we do as scientists. And I've spoken to a number of other editors, some of whom are here, and everybody's having a much harder time these days finding reviewers for papers. And there may be many reasons for that, but I'd really encourage all of you to resist the temptation to hit the delete button when you get an invitation to review a paper, because it really is important. And kind of along these lines, one additional thing that I've agreed to do is Cold Spring Harbor Labs is has asked Jonathan Karn and myself to edit a volume on HIV protocols. So if you have HIV-specific protocols that you think might be suitable for this volume, please come talk to me or send me an email. Okay, so what are we doing now? Things are going to get a little bit dry and a little, sciencey, but this is going to be a little bit light, not a lot of data. But I just want to tell you a little bit about what I'm excited about right now. And so current projects, I'm not going to list them all, but some of the key projects, antagonism of viral envelope glycoproteins by host factors, including the March E3 ubiquitin ligases and GBB5. And I'll ask you to please attend Hannah's poster this evening, number 232, about GBB5. For many years, we've been interested in developing HIV-1 maturation inhibitors. I'll say a little bit about that in a moment. We've also been working to elucidate the role of IP6 in virus replication in close collaboration with Leo James. This has been led in the lab by Alex and postback Bin. Alex will be giving the Andy Kaplan talk Saturday morning at 9:30. So I hope you'll all drag your hungover selves out of bed and attend that talk. You heard yesterday from Yuta Akichi about mutations in HIV-1 envelope and the nucleocapsid domain of GAG that reduce the susceptibility of HIV-1 to antiretrovirals. We've also become interested in the role of the virion lipid composition in HIV-1 maturation. This has been led by Wahid in the lab, joined recently by postdoc Needy and postdoc Lindsay, who I misspelled your name. I'm very sorry. I'm never going to hear. I'm never going to hear the end of this. She gave a poster the other day, which hopefully many of you saw. We've also been elucidating resistant pathways for integrase transfer inhibitors in terms of integrase, and also studying matrix structure and envelope incorporation. This is work in collaboration with Pedro and Zhang, and there's a poster on this also this evening. Okay, so for the rest of the science part of the talk, I'm going to focus on the last step in the virus replication cycle, namely virus maturation, which, as all of you know and we'll hear much more about tomorrow, is the conversion of the immature particle to the mature particle that contains this condensed conical core. So the immature particle contains this immature GAG lattice. Through the action of the viral protease cleaving GAG and GAG-POL, there's this major morphological transformation of the virus particle, the most striking aspect of which is the formation of the mature conical core, which contains an outer layer of CA protein, known as the capsid, and inside of which are the two single-stranded copies of the viral RNA genome in complex with RT and integrase. And so on this slide, we're going to do a deep dive into the structure of the immature GAG lattice, focusing specifically on a region of the immature lattice spanning the C-terminus of capsid and spacer peptide 1. This region folds into a 6-helix bundle that's depicted here, again spanning the junction between capsid in gray and SP1 in orange. And this 6-helix bundle contains deeply buried within it the last cleavage site in the GAG processing cascade, namely the cleavage site between capsid and SP1. So this 6-helix bundle can be likened to the, boy, here I go with drinking again, can be likened to the stem of a wine glass with the stem holding together the 6 monomers of the GAG hexamer. And if we look at the 6-helix bundle in a top-down view, we see that it has a central channel, which turns out is the binding site for IP6, but also for maturation inhibitors. And I'll say a bit more about that in a moment. So this is a cartoon depiction of the GAG processing cascade, which terminates or ends with the separation of spacer peptide 1 from CA, giving rise to mature CA. So more than 20 years ago, together with our collaborators at Panicos Pharmaceuticals and independently in Chris Aiken's lab, it was shown that the first-in-class maturation inhibitor, Beverimat, acts by blocking this last step in the GAG processing cascade. And we can visualize this on gels such as the one shown here, where in the absence of beveramat or now other maturation inhibitors, we see almost entirely mature CA, whereas in the presence of beveramat or other maturation inhibitors, we see this accumulation of capsid SP1, this intermediate here. So this can be very, very carefully quantified. And if we look at the virions produced from maturation inhibitor-treated cells, we see that unlike the virions from control cells that have these nice conical capsids highlighted in red, the virions produced from maturation inhibitor-treated cells contain this crescent of electron density that represents a stabilized remnant of the immature GAG lattice. So, I'm leaving many years to work out, but I'll just leave this section with a description of how maturation inhibitors actually work. So, I've told you that the capsid SP1 junction folds into a 6-helix bundle. This is a very dynamic region of GAG that's in an equilibrium between the 6-helix bundle conformation that favors virus assembly and a more disordered or unfolded state that favors maturation, because the cleavage site, which is buried in the bundle, is accessible when this region unfolds. Maturation inhibitors, as I mentioned, bind inside the 6-helix bundle, and they stabilize the bundle by binding at that site. So maturation inhibitors shift the equilibrium to the right, preventing cleavage by protease at the capsid SP1 junction. Now, when we selected for maturation inhibitor resistance, we often found mutants that were dependent on maturation inhibitors for their replication. These mutations shift the equilibrium to the left, disordering the bundle and causing severe defects in virus assembly. These defects can then be rescued by addition of the maturation inhibitor, which explains why they're maturation inhibitor dependent. And if we propagated these maturation inhibitor dependent mutants, we picked up compensatory mutations that acted like maturation inhibitors in stabilizing the bundle, the best known of which is SP1T8I. So in the context of these maturation inhibitor dependent mutants, T8I will reestablish the native equilibrium of this region of GAG, allowing the virus to be replication competent and fully infectious. So take-home messages: the CASP1 6-helix bundle is highly dynamic and is a structural switch between assembly and maturation. Maturation inhibitors bind inside the 6-helix bundle, stabilizing the bundle and preventing access by the viral protease. So if we look at the capsid SP1 junction region across different subtypes of HIV-1, we see that this region is highly conserved, and the residues to which we mapped beveramat resistance mutations are also highly conserved. However, downstream in SP1 is a region that's much less conserved or more polymorphic. And one of these polymorphic residues that's particularly common in some subtypes of HIV-1 is what we call V7A, so an alanine instead of a valine at residue 7 of SP1. So beveramat performed well in many clinical trial participants, but the activity of beveramat was severely compromised by polymorphisms in SP1, such as V7A and a few others. So together with Heterodrugs and DFH Pharma, we've developed highly potent and broadly active beveramat analogs that overcome the loss of susceptibility to beveramat imposed by these SP1 polymorphisms. So on the left is the structure of the first-in-class maturation inhibitor beveramat, and then one of our lead compounds, and we screened hundreds of these molecules, one of our lead compounds, which I'll refer to as HRF4467, it contains modifications at 3 positions around the beveramat scaffold. And I'm not going to show you data from hundreds of analogs, but just a little snippet of data to convince you that this particular second-generation maturation inhibitor is able to overcome the block in beveramat activity imposed by polymorphisms like V7A. So here's a capsid SP1 processing assay using gels like the one that I showed earlier. So beveramat in red shows essentially no activity against the V7A polymorph, whereas the analog of beveramat shows very high potency in blocking capsid SP1 processing. We can also look at this in an antiviral assay using single-round infectivity and allowing us to calculate antiviral IC50s. So looking at wild-type NO4-3, comparing beveramat and HRF4467, we see that the IC50 of beveramat is about 270 nanomolar. The IC50 of 4467 is about 5 nanomolar. Much more importantly, against V7A, we see that the dose-response curve for beveramat is essentially flat, that is, no antiviral activity, whereas 4467 essentially does not lose any antiviral activity against V7A, 5 nanomolar, IC50, pretty much the same as what we saw with wild-type NO4-3. And I'll just tell you, without showing you the data, that the increased potency of 4467 relative to beveramat extends to other polymorphs and diverse HIV-1 subtypes. So we've collaborated with Tatiana Polanova's group, who saw the structure of beveramat bound to the 6-helix bundle by solid-state NMR. This was published last year. And a few key takeaways from this work. The orientation of beveramat in the 6-helix bundle was unequivocally determined. This is important in terms of doing the structure-based drug design. It's important to know what the contacts are and how this compound is oriented. As has been inferred from previous studies, beveramat binding stabilizes the 6-helix bundle. The maturation inhibitor resistant mutant A1V shows no binding to beveramat, but the V7A polymorph that I described binds beveramat, but without a stabilizing effect on the 6-helix bundle. And structures of our second-generation maturation inhibitors, including 4467 that I just told you about, are currently being determined. So ongoing work on maturation inhibitors. We're continuing to develop more potent maturation inhibitors and moving them into preclinical and clinical studies. This is being done primarily with DFH Pharma. With Oakcrest Institute, we're developing long-acting maturation inhibitor formulations. We're continuing to define determinants in GAG that influence susceptibility to maturation inhibitors by testing antiviral activity against a range of viral isolates and selecting for resistance. And with Tatiana Polanova and her colleagues defining the structural correlates of maturation inhibitor activity. So I'd like to show this slide because I think this work and similar studies illustrate how basic molecular virology can lead to the development of novel antiviral drugs, but also how studying these novel drugs can feed back and inform basic aspects of molecular virology. So I've been telling you about the 6-helix bundle. It turns out that the natural ligand for the 6-helix bundle is, of course, not maturation inhibitors, but IP6, which sits at a position high up in the bundle, just on top of or just above where maturation inhibitors bind. This is a kind of a cartoony structure of IP6. It's this highly negatively charged molecule. And what's quite fascinating, I think, is that IP6 binds a completely different site in the HIV-1 virion that is at the center of the mature CA hexamer or CA pentamer. And so IP6 binds, stabilizes, and promotes the assembly of both the immature GAG lattice and the mature capsid core. And I don't have time to really go through all the work, and Alex Kleinpeter is going to talk about this in much more detail on Saturday. But I'll just show this cartoon of how we think IP6 functions in the HIV-1 replication cycle. So IP6 is denoted as this little asterisk, which is present at fairly high concentrations in cells. So during virus assembly, IP6 binds the center of the 6-helix bundle, stabilizing the bundle and promoting the assembly of the immature GAG lattice. When this immature GAG lattice is destroyed by the viral protease during maturation, IP6 is released from this binding site and then able to bind at the center of the CA hexamer or pentamer, allowing the formation of the stable capsid. We believe that this IP6 remains associated with the mature capsid during its journey to the nucleus. So here are just a number of papers that we've published on this topic, again in close collaboration with Leo James, and I certainly need to point out that a number of other groups, many of whom are represented here, have contributed to this model for IP6 function. So again, I'm kind of harping on this, but I really want you guys to come to that talk. And I know Saturday morning can be a challenge, but it is 9:30 and not 9, so we get a little bit of a break. Okay, so I mentioned earlier that when Akira Ono was in the lab, he showed that HIV-1 assembles in lipid RAF microdomains in the plasma membrane, and that PIP2 is an important determinant in taking GAG to the plasma membrane. Consistent with the idea that HIV-1 assembles in lipid RAF microdomains, the viral envelope is enriched in RAF-like lipids like cholesterol and sphingomyelin. This raises interesting questions about the role that virion lipid composition plays in the late stages of the HIV-1 replication cycle. And we approach this question by altering the lipid composition of HIV-1 virions by treating virus-producing cells with inhibitors of the lipid-modifying enzyme neutral sphingomyelinase 2, or NSMase2. These inhibitors were obtained from our collaborators, Norm Howie and Barbara Slusher, at Johns Hopkins University. So this is one of the early experiments that we did when we started working with the inhibitor. This is the structure of one of these inhibitors, PDDC. So what we observed is this is Western blots of cell and virion-associated material from cells treated with an escalating concentration of PDDC. What we observed is that as we increased the concentration of this NSMase2 inhibitor, we saw a very, very pronounced and dose-dependent inhibition of GAG processing, such that at the highest concentration we saw very little P24 in either the cell or the virion fraction. A inactive variant of PDDC, which we call control or control 5, shows no such activity in this assay. Now we know that GAG processing is essential for virus infectivity, so we weren't particularly surprised to see that treating virus-producing cells with PDDC caused a very severe loss of infectivity of the virions produced from those cells. So to avoid conclusions that were based on off-target effects of PDDC, we inhibited the activity of NSMase2 by a different approach, namely knocking it down with siRNA. And we saw essentially the same thing. So in the presence of non-targeting siRNA, we see robust levels of P24 being generated, whereas in the presence of the NSMase2 specific siRNA, we saw a loss of P24 in both the cell and the virion lysates. And again, virus infectivity was very severely compromised. So these results demonstrate that NSMase2 in the virus-producing cell is required for GAG processing. We, of course, looked at the resulting virions by thin section EM and also cryo-EM with the help of Pedro Zhang. So in the thin section EM untreated cells, you see these very nice conical capsids, typical of mature infectious virus particles, whereas those viruses produced from cells treated with the NSMase2 siRNA or PDDC were uniformly immature in their morphology. That is, they were sort of like donuts rather than having condensed conical capsids. We also saw a number of these intriguing little membranous blebs and regions of large discontinuities in the immature GAG lattice. So disrupting NSMase2 in virus-producing cells with either a small molecule inhibitor or siRNA severely impairs HIV-1 GAG processing and blocks virion maturation and infectivity. The requirement for NSMase2, and I'm not showing you the data for this, in virus maturation appears to be specific for primate lentiviruses. So if we look, for example, at MLV or RSV, we see absolutely no effect on either GAG processing or particle infectivity. PDDC suppresses HIV-1 replication in humanized mice infected with HIV-1, and interestingly, this suppression of viral loads continues even after removal of the inhibitor, in contrast to what we see with standard antiretrovirals, where there's a very rapid rebound after discontinuation of the inhibitors. So the take-home message here is that NSMase2 is required for the morphogenesis and maturation of primate lentiviruses. Now, in some unpublished work, we've found that resistant selection experiments with PDDC and analysis of chimeras between a sensitive virus like HIV-1 and an insensitive virus like MLV suggests that protease activation, perhaps protease dimerization, is the step that's inhibited by NSMase2 disruption. So just to kind of show this, what we're thinking is a working model in this cartoon fashion. So all of you, I think, know that levels of GAG-POL are present in HIV-1 or retroviruses at levels that are much lower than those of GAG, about 5% in the case of HIV-1. We also know that protease is an obligate dimer, so GAG-POL must dimerize in order for protease to be activated and GAG to be cleaved and virus maturation to take place. So we think that possibly by changing the lipid composition of the viral membrane, we're inhibiting or interfering with the activation of the viral protease, perhaps by preventing GAG-POL dimerization. And these models are currently being tested in the lab. Okay, this concludes the science part of my talk. So just to preempt the question of what advice do you have for us, I'm going to list a few pieces of advice for students or maybe some of the younger postdocs. Maybe some of the senior people too, I don't know. Ask important questions, not the easy questions. Be passionate about your work. Scientific research is one of the most impactful and exciting human endeavors. Do your part to contribute to the service aspect of our profession. I already touched on this. Encourage and promote diversity in science. We still have a long way to go. Respect and cherish your colleagues. So the people you're working with now in your labs, the people you meet at meetings like the one here, will be your close friends and colleagues for the rest of your career. So I think it's really important that you develop those relationships. And I do a little bit of bike racing, and usually before a race, the race organizer will give a little spiel about, when you're out there on the race, watch for cars, make sure you stay hydrated, so on and so forth. But then the parting piece of advice that the race organizer has, which I think is also relevant to science, is don't be a jerk. Okay, I've come to the acknowledgments part. I'd like to thank Stephanie in the meetings office for providing me access to all those great old archival photos. I'd like to thank my former mentors, David Shalloway, Rex Risser, Howard Temin, Malcolm Martin. I'd like to thank my current lab. It's really a phenomenal group of young scientists. They're a lot of fun to work with, even though Lindsay will never forgive me for misspelling her name. I'd like to acknowledge my colleagues in the HIV DRP. They're a wonderful group of people to work with. My current collaborators, this is I think the full list. I hopefully didn't miss anybody who's in the auditorium. And in red, I've highlighted those people who are working with us on the work that I actually talked about. And a number of important former collaborators, funding sources, former trainees. The list gets longer and longer as the years go by. And I'd also like to take a moment to remember the people who were regular attendees of this meeting who are no longer with us. My PhD advisor, Rex Risser, Andy Kaplan, Ute von Schvedler, and David Dursey. These were all photos taken here at Cold Spring Harbor. And in particular, I would like to remember Judith Levin, who many of you remember as a regular attendee of this meeting. She was a lovely person and a very creative scientist. If you want to learn a little bit more about her life and work, I'll direct you to this short tribute that Corinne, Ellen, and I wrote about her and published in Viruses early this year. And I would like to dedicate this talk in her memory. So with that, I'm happy to take any questions. Now you guys are going to make me cry. Come on. Questions?
Maybe we can start off with a question online. So, anonymous attendee is asking.
Geez. Oh, wow.
I don't know if this is going to be a critical question. So, does IP6 have any role in the formation of a cone-shaped capsid instead of a fully icosahedral sphere?
Yeah, so IP6 is especially important for the formation of pentamers. And so in order to have appropriately shaped capsids that are functional in the next round of infection, IP6 plays a crucial role because pentamers are key players in the shape of that capsid.
So, Eric, no surprise, that was absolutely wonderful.
Thank you.
So,
not as good as yours, but...
So to me, you have always been the picture of a person calm and in control and unfrappable throughout the wonderful things that you've done.
Boy, I wonder if my wife is listening.
So, as you've traversed through just about the entire replication cycle, has there ever been a portion of it that you studied where you said, oh, God, I can't figure this out?
There are lots and lots of things that I've not figured out. And, you know, most of those are recent things. You know, you heard Yuta yesterday talking about these NC mutants, which we still don't fully understand. I think they're very interesting, exciting. There are some old work that we did on some matrix mutants that didn't make sense that I think now are beginning to make sense with the help of Peiju and Zhang. We're doing some structural work that I think is shedding some of the secrets of those mutants. So I think, you know, I've always done new things, but in the back of my mind has always been the things I did earlier in my career. And, you know, there are things that we have to put on the back burner because we don't understand them, we don't have the technology to really probe, you know, ask the right questions. But we can then revisit those over time with new technologies, cryo-EM being, you know, one example. So I think that it's always fun to kind of go back to old questions and revisit them.
I sort of have a follow-up to Carol's question. I'll never forget once you came up here and you showed data for mutations of every single residue in matrix. And I remember everyone was just sort of like, I don't know.
It wasn't.
So is there a residue in the virus you haven't mutated?
I think there are 2 or 3 we're working on this.
I have a question related to PDDC. How do you explain this long-term control after treatment?
Yeah.
Is tissue concentration something about the half-life on tissues versus blood?
Yeah, so, you know, I'm always really hesitant to claim that PDDC is reducing the size of the reservoir because that's the holy grail of cure research, and I just don't want to go there. But PDDC might be reducing the size of the reservoir. So our collaborator, Norm Howie, and actually there's a poster that I believe is tonight that digs into the observation that PDDC selectively kills HIV-infected cells. And so that would be a mechanism that would explain either a lack of rebound or a delayed rebound in these treated animals. But we need to do a lot more work to really pin that down.
Talking about mechanisms here,
Yes,
sorry.
yes,
Do you think drinking helps to create good models? Because you had your wine glass, I've seen some other similar things.
Yeah, I'm a big proponent.
Eric, that was a great talk. Very inspiring, I'm sure, for all of us, but especially for the young folks who don't know all of that, haven't seen all those pictures. So
they were fun to look at.
One thing, you mentioned service, but one part of that I'd like you to comment more on. I've had the chance to see how your leadership works in the dynamics and replication program and hear from some of the colleagues about that. You kind of glossed over that, but I know you've done a terrific job there. And so what do you think your leadership style is that allows a group of very different scientists to get along and do great work together?
So I don't want to take any credit for that. And the reason is that the DRP is a fantastic group of people. They're a pleasure to work with. And so it's been easy. I'll give you the real answer later.
So, first, congratulations on a really spectacular body of work.
Was that really you in that picture, by the way?
It might have been. I guess my question is for you as sort of one of the leaders, intellectually and otherwise, of our field. What do you see for the future of retrovirology?
Yeah, so I guess this kind of touches back on what I was saying a moment ago, which is, you know, when you sit around with people and you start talking about retroviruses and their replication, you really quickly realize that there's a lot we don't understand. So we know the basic stuff. And, you know, we did at the beginning, you've been at this meeting a number of years too, and we saw this cartoony, you know, depiction of the replication cycle. We do know a lot, no question about that. But, you know, when you really get a couple layers in and you realize the knowledge ends. And so I think, you know, there's still a huge amount to be learned. And I think as technologies improve and become more sophisticated, we're going to continue to reveal, you know, new secrets. And, you know, it's great for me to see so many people in this room. There are those out there who will say, well, HIV is done and, you know, we ought to be studying SARS-CoV-2 or whatever else. Neurodegeneration, Alzheimer's, and those are very important things too. But there is still a lot we don't know, and I think there's still a need for understanding HIV in greater detail.